Hi, this is Sir Steven, and today, as a part of Medical Microbiology series, we will be talking about microscopy and in vitro culture. Microscopy in general is used in microbiology for two basic purposes. The first one is to detect microbes and the preliminary or definitive identification of your microbes. The microscopic examination of your clinical specimens is used to first detect bacterial cells, second, some fungal elements, third, parasites. It could be on the eggs, larvae, or adult forms, or clumps of viruses, which are called, or which is seen in the microscope as viral inclusions that are present in these infected cells. Also with morphologic properties, which are going to be used as a preliminary identification of your most bacteria, and these are also used for the definitive identification of many fungi and parasites. The microscopic detection of organisms stained with antibodies are labeled with the fluorescent dyes or other markers that has proved to be very useful for the specific identifications of many of your microorganisms, clinically important microorganisms. These are the 5 general microscopic methods that are being used: bright field light microscope, dark field microscope, phase contrast microscope, fluorescent microscope, and lastly, the electron microscope. And we will be discussing all of these microscopic methods in this presentation. You can see here in this image is an optical microscope that are used in a wiki science competition in Thailand. You can see and also review some of the basic parts of the microscope as noted in this typical optical microscope that are used in a BSL-1 laboratory or BSL-2 laboratories. So the image has been numbered for your convenience, and you should be able to identify and know the functions and parts of your typical optical microscope. The next image is showing you a 2 Leica oil immersion microscope objective lenses. On your left is a 100x microscope, and on your right is a 40x magnification lens or objective lenses. I am showing you some of the illumination of your different microscopes. For this photograph, it is coming from a bright field illumination. Sample contrast comes from the absorbance of light in the sample. The second image is another type of micrograph. This is called cross-polarized light illumination. The sample contrasts from rotation of polarized light through the sample. This is a dark field illumination. Sample contrast comes from light scattered by the sample. This is phase contrast illumination. Sample contrast comes from the interference of different path lengths of light through the sample. This is more you can see the minute details within the structures. The next image is showing you a red blood cells that are seen by the dark field microscopy. This has a magnification of 1000x. Showing you the diagram of how the light path travels in the dark field microscope by Richard Wheeler in 2006. This is the diagram. On yellow, only the scalar light is transmitted. There is what we call as your direct illumination block. Some light is scattered by the sample. This is your sample. We have also here a condenser lens and a light source. And the difference is there is a dark field patch stop on this dark field microscope. Typically, this is only used in some laboratories. An image will show you such an example of a dark field microscope. These are micrographs of Baker's yeast or the Saccharomyces cerevisiae in a DIC microscopy. This is performed with the Olympus BX61 microscope, showing the budding of your Baker's yeast. Also with the next image, these same cells on your left and your right is imaged by traditional bright field microscopy. This is the typical one we're using in the laboratory on your left, and on your right is a phase contrast microscope. See the difference. This is an upright fluorescent microscope, the Olympus BX61, with a fluorescence filter cube turret above the objective lenses. You can see this black object here that are coupled with, on top of it is a digital camera. This is a human lymphocyte, the nucleus of a human lymphocyte stained with a DAPI with chromosome number 13 in green and 21 in red. The centromere probes are hybridized by fish. This is also called as your fluorescent in situ hybridization. Next micrograph is a yeast cell membrane, visualized by some membrane proteins that are fused with RFP, the red, and the GFP, the green fluorescent markers. So the imposition of light from both of the markers will show the yellow color, the mixture of the red and the green fluorescent markers. This is another microscope, which is the last microscope that are to be discussed, a modern transmission electron microscope. And the diagram here will show you how the electron microscopes are operating. This is more intensive process on how how physics works in electron microscopes. But we will not gonna talk about that one. I'm just showing you how light is being scattered using these electron beams hitting the sample, and some of the background, okay, of the physics of microscopes. With the emergence and the accessibility of your electron microscopes, we're able to see the first image of the Bacillus subtilis taken with a 1960s electron microscope. You can see here how detailed the structure of these bacterium. With our next processes in medical microbiology using your microscopy and in vitro cultures, we will gonna talk about direct examinations. One good example of that is a blood smear from a pathological examination. This is what we call as your wet mount. A blood sample, okay, blood smears. Second one is your KOH or your potassium hydroxide. This material or this compound is used to dissolve proteinaceous material and facilitate the detection of your fungal elements. Later on, we'll gonna see how KOH is being used in medical microbiology. With direct examination, this is also used. India ink. This photomicrograph depicts the Cryptococcus neoformans using a light India ink staining preparation. These are life-threatening infections that are caused by your encapsulated fungal pathogen. We call it the Cryptococcus neoformans, that have been increasingly steadily over the past 10 years because of the onset of your acquired immunodeficiency syndrome and expanded use of immunosuppressive drugs. Next is direct examination of your 2% Lugol's iodine solution drops thyroid support supplement, 2 ounce. Okay, this one, this iodine is being added to wet preparations of your parasitology specimens. This is also to enhance the internal structures and contrast their um, inside structures. Okay, this also this compound here is also allows us to facilitate differentiation of your amoeba and your host white blood cells. Another method is in the form of your differential stains. In your general microbiology laboratory, you've also learned the importance and the processes that are present in the gram staining of your bacterium, your fungi. This is a photomicrograph of a gram positive. It's pretty obvious in this image that this is a gram positive because of the dark purple or dark violet color, your streptococci that are being surrounded by pus cells. Another photo of a differential stain, a gram stain, the same thing as with the previous slide. This is the Candida albicans from a vaginal swab. The patient has experiencing candidiasis, and the medical technician were able to isolate these small oval chlamydospores that are 2 to 4 micrometer in diameter. These are fungal cells, and all fungal cells will show a gram-positive result using the gram staining method. With differential stains, this is also used. We call it the iron hematoxylin stain specimen. This is revealing us the presence of a mature entamoeba cyst which contained 5 visible nuclei. These are the nuclei that are found in the middle, each with its eccentrically located choristome and a peripherally located chromatin. This is another differential stain with a silver stain. We also call it the G-methenamine silver that are demonstrating a fungus. This is histoplasma that are morphologically seen as black round balls in a liver biopsy. Okay, this one in general is performed in histology laboratories rather than in microbiology laboratories. This is also used as a primary stain for detection of these fungal elements in your tissue. In this example is a liver biopsy. Although other organisms in this examination is, let's say for example, the bacteria can be detected also, because silver staining requires skill. With that, non-specific staining can render slides that are incapable of being interpreted. So it could also give us some characteristics or visible colonies of your bacteria. This is a toluidine blue stain in your vasculitic peripheral neuropathy. This is primarily used for the detection of pneumocystis organisms in your respiratory specimens. Cysts stain reddish blue to dark purple in a light blue background. You can see that there are some cells that are found in this photomicrograph. Background staining is removed by sulfation reagent. Yeast cells stain and are difficult to distinguish from your pneumocystis cells. Trophozoites do not stain, and in this, many laboratories have replaced this stain with specific fluorescent stains. This is your pneumocystis jirovecii cysts from your bronchoalveolar lavage. these are stained with your toluidine blue O stain. Okay. Next is a trichrome stain. This is just a fancy photomicrograph that I got from the internet and the research from Garcia et al. In the first row, these are the permanent stain options for various organisms. The first row, we identify them from the left to right. Wheatley's trichrome stains of first is your Giardia lamblia trophozoite. Second is an Entamoeba histolica trophozoite with ingested red blood cells. Third is an Entamoeba coli cyst with red chromatoidal bars, and your Chilomastix mesnili cysts in the second row. These are stained with iron hematoxylin stains. Okay, we are just much concerned with the first row because they are stained with the trichrome stain. Right Gamsa stain, also a part of a differential stain. Well, right Gamsa stain is used to detect blood parasites, viral and chlamydial inclusion bodies, and Borrelia, Toxoplasma, Pneumocystis, and Rickettsia species. This is also known as your polychromatic stain, which is containing a mixture of methylene blue, azure B, eosin Y. The Gamsa stain combines methylene blue and eosin. Eosin ions are negatively charged and stain basic components of cells orange to pink, whereas other dyes will stain acidic cell structure in shades of blue to purple. Protozoan trophozoites have a red nucleus and grayish-blue cytoplasm. Intracellular yeasts and inclusion bodies typically stain blue. Rickettsia, Chlamydia, and Pneumocystis species stain purple. That's why this is in this micrograph, we can see the internal nuclei of the cyst up to 8 in number, and this is the Pneumocystis jirovecii in the Gamsa stain of BAL. Another type of staining is what we call as your acid-fast stain. This is used to stain mycobacteria and other acid-fast organisms. Organisms are stained with basic carbonyl fission, and they resist decolorization with acid-alkali solutions. Your background here is counter-stained with methylene blue. Your target organisms will appear red against the light blue background. Uptake of your carbonyl fission requires heating specimen, or we call that the hot acid fasting. In this technique, this is called the Ziel-Nielsen stain that visualizes your Mycobacterium tuberculosis. On the other hand, the cold acid fast stain, or also known as your Quinoan method or Quinoan stain, developed by Joseph Quinoan. They both share the same principle with the Ziel-Nielsen stain, but in this method we don't use heat. Rather, this is also called as the cold acid fasting, showing you images of your acid fast positive in red and acid fast negative in blue. This image is the another staining, acid fast stain, using the auramine rhodamine stain. This is an oocyst of your Cryptosporidium parvum stained with the fluorescent auramine rhodamine stain. With acid fast staining, there is also what we call as your weak decolorizing agent, or the modified acid fast staining method. This is to identify rapidly the detection of your Mycobacterium tuberculosis. In this method, a weak decolorizing agent is used with many 3-acid fast stain listed, whereas the Mycobacterium has been noted as a strongly acid fast. Other organisms in this method will stain weaker, such as your nocardia, rhodococcus, chucamarella, gordonia, cryptosporidium, isospora, sarcocystis, and cyclospora. Because of that, these organisms can be stained more efficiently by using a weak decolorizing agent, and organisms that retain this stain are referred to as partially acid fast. Another staining method in the method of fluorescent stains, this is using an acridine stain, showing you the corneiforms bacteria. Next image is the fluorescent staining of yeasts with carclofluorowhite. The cell walls of your yeasts will show you a vivid blue color. This is used, the calcofluorowhite is used to detect fungal elements and pneumocystis species. Stain binds to cellulose and chitin that are found in the cell walls of your fungi. Microscopists can mix the dye with potassium hydroxide, because a lot of the laboratories have been replacing the traditional potassium hydroxide with this staining. This is a direct FA stained of your mouse brain impression smear that are revealing the presence of the bacterium Chlamydia pseudomonas. These are such as your antibodies. Okay, we stained the antibodies. It can either be the monoclonal or a polyclonal. Your antibodies are complexed with fluorescent molecules, and specific binding to an organism is detected by the presence of microbial fluorescence. This technique has proved useful for the detection and identifying many organisms such as your Streptococcus pyogenes, Bordetella, Francisella, Legionella, Chlamydia, pneumocystis, Cryptosporidium, Giardia, influenza virus, herpes simplex virus. The sensitivity of this test, direct FA, is determined by the number of organisms present in the test sample and the quality of the antibodies used in the reagents. This is a blood agar, okay? A part of your non-selective culture media or the generalized culture media. It will allow the growth of many or almost all microorganisms. In the recovery of fungi and bacteria, this is also used. These are red blood cells on an agar plate that are used to diagnose infection, the blood agar. On the left is a positive staphylococcus infection, and on the right is a positive streptococcus infection, showing you the differences in how the microbes are utilizing the presence of the blood, okay, in these agar, because they were able to perform hemolysis. Another non-selective culture media is in the name of chocolate agar, showing your Francisella tularensis colonies. In chocolate agar, this is also used for the recovery of some of your bacteria, including your Haemophilus and your Neisseria gonorrhea. Another non-selective culture media is the colonies of your Burkholderia pseudomalae and MHA or Muller-Hinton agar after 72 hours of incubation. Well, MHA is used for bacterial susceptibility test. Another non-selective culture media is in the name of the thioglycolate broth. This is recommended to isolate strict anaerobes should an anaerobic infection be suspected. These anaerobes don't need the presence of strict anaerobes don't need the presence of your O2 in the atmosphere. That's why it's tightly sealed, and we're using this thioglycolate broth medium. Another non-selective culture media is your Sabrod dextrose agar. Your Sabrod agar is a type of agar that growing or allowing us to cultivate dermatophytes and other types of fungi because of the peptones that are containing in this medium. This can also grow filamentous bacteria such as your nocardia. It is highly utilized for research and clinical care. Another type of media is a selective and differential culture media, a McConkey agar plate with an active bacterial culture. This is the Proteus vulgaris. Your McConkey agar plates are selective for gram-negative bacteria. So only the gram-negatives are growing in this medium. And they are differential for lactose fermenting species and non-lactose fermenting species. Here is an example of your MSA plate, also known as your mannitol salt agar plate. These are numbered in this diagram. These are again selective for staphylococci and differential for the presence of your Staphylococcus aureus. Micrococcus species number 1, Staphylococcus epidermidis number 2, and Staphylococcus aureus colonies in number 3, showing you the bright yellow colonies in this plate. Another selective differential culture media is your XLD agar, showing you a salmonella growing here in your XLD agar. These are selective differential agar for salmonella and shigella in enteric culture. So when you say enteric, these are coming from your gastrointestinal tract. Next is your Lowenstein-Jansen medium. We can see in this image a distinctive clusters of your colorless Mycobacterium tuberculosis because this medium is highly selective for mycobacteria. Middlebrook's tube slant agar is also selective for mycobacteria. Crumb agar, showing you colonies. These are selective, differential for selected bacteria and yeasts. And luckily, we're able to see the crumb agar and their website showing us what these colorful colonies signify. First, this dark pink to reddish, their appearances is denoting of an Escherichia coli, turquoise blue for Enterococcus, brown halo for Proteus, metallic blue for Klebsiella, Enterobacter, and Serratia, golden opaque small for S. aureus, metallic blue with red halo for Citrobacter, pink opaque and small for S. saprophyticus, colorless for Candida albicans, light blue for Streptococcus agalactiae, and translucide cream to blue for Pseudomonas aeruginosa. Next is your IMA or the inhibitory mold agar. These are moderately selective medium used for the isolation of your pathogenic fungi, selective for your molds. BCYE agar or the black charcoal agar is useful, as you can see the color is black, for the recovery of your Legionella and Nocardia species. Cysteine tellurite agar, also in crimson red color. This remilk cysteine tellurite blood agar is a solid medium that are useful for the qualitative procedures for the isolation and cultivation of your Corynebacterium diphtheriae, the one that causes diphtheria. The peptone that are found in this medium will provide nitrogen and carbon compounds, amino acids, and trace minerals, which are very essential for the bacterial growth. Lymph broth is um, for the selective enrichment of your group B streptococcus, such as your streptococcus agalactiae from your clinical specimens. Lymph broth is highly selective or specialized for the recovery of your streptococcus agalactiae. McConkey sorbitol agar. This is also specialized for the recovery of your Escherichia coli strain 0157. This particular strain of Escherichia coli will cause severe intestinal infections, especially in humans. And this is the most common strain that causes illness in people. This is highly differentiated from other E. coli because of the production of a potent toxin that damages the linings of your intestinal walls and causing your bloody diarrhea. And they call the bloody diarrhea as an enterohemorrhagic E. coli infection. The CDC, or the Centers for Disease Control and Infection Prevention, reports about 70,000 cases of this E. coli infection that occur in the US each year. Regan Lowy agar is also black, same with the BCYE. This is useful for the recovery of your Bordetella pertussis. TCBS agar plate is useful for the recovery of your Vibrio species. On your left is your Vibrio cholerae, and on your right is your Vibrio parahemolyticus in green. Another technique is being discovered in 1949 by this person. His name is John Franklin Anders. He described before 1949 how to cultivate mammalian cells for the isolation of your poliovirus. This technique has been expanded for the growth of most strict intracellular organisms. The cell cultures can either be cells that grow and divide on the surface, for example, the cell's monolayer, or grow suspended in broth, because that's the characteristic of a normal cell. Okay, if your cells are growing above the monolayer of the cell, that's not considered to be a normal cell. It could either be a cancerous cell, okay, or a tumor. Some cell cultures are well established and can be maintained indefinitely. These cultures are commonly commercially available. This is a bottle of a DMEM cell culture medium with a schematic representation of a 2D culture, a 3D culture, organ on a chip, and an in vivo study. These are on the arrow shows you significantly of higher physiological relevance. And these are the references that are being used in this presentation. If you have questions regarding this lesson or this presentation, feel free to comment down your suggestions or feedback and questions below. Again, thank you so much for listening. And up next, like all the evidences that are being left at the scene of a crime, your deoxyribonucleic acid, your DNA, or your RNA, or some of the proteins of your infectious pathogen in your clinical samples can be useful to identify the causative agents. And this is a part of the next series of your medical microbiology called the molecular diagnosis. Again, have a wonderful day and stay safe.